Hey all, Mike Fave here. I'm going to go through another study today. This one is about fish oil and its effect inside colitis. The title of this study is Fish Oil 1008's Omega-6 Polyunsaturated Fatty Acid-Induced Dysbiosis and Infectious Colitis, but impairs lipopolysaccharide dephosphorylation activity, causing sepsis. So the gist of this study is they had different groups. They were fed high omega-6 or high omega-3 fatty acids, and then they did a bunch of tests on them, and then the rats were essentially infected with a pathogen here called Citrobacter rodentium, and then the researchers wanted to see what happened to the rats. It's quite an interesting study. I think the outcome is going to be probably not what everybody would expect, considering all the hype behind fish oil. But anyway, let's jump into it. I'm going to start here with the introduction. I'm not going to read the entire study, but I'm going to pick out important pieces. So researchers start out here. They say we recently demonstrated that intestinal microbiota protects against lethal infectious colitis by regulating protective inflammatory responses and plays a major role in modulating pharmacological efficacy of drugs to attenuate colitis. However, little is known about the effects of dietary lipids on inducing specific microbial changes that are either protective or detrimental in IBD. Essentially what they're talking about here is that the intestinal microbiota, so your microbiome, can protect against inflammation of the intestine, particularly the colon. So altering the microbiome with things like probiotics, prebiotics, foods, all that type of stuff, different fibers, can protect against infectious colitis. There's a lot of research about this for different supplements, different compounds, different foods, etc. And so what they wanted to see here was they wanted to see if different lipids in the diet, specifically different types of polyunsaturated fatty acids, will have an effect as well. So that's what they set out to study here. They go on here and they're going to talk about the lipids. They say in the past few decades, the lipid content of the Western diet has shifted from saturated fats to predominantly cardioprotective, in quotations, omega-6 polyunsaturated fatty acids, of which the intake has risen by 54% over the last 3 decades in Canada. Such benefits of omega-6 PUFAs are currently in question as new evidence reveals the detrimental effect of excess omega-6 PUFA intake in chronic metabolic disorders. The specific impact of PUFAs on gut health are less clear. However, consumption of high-fat diets, and specifically diets high in omega-6 PUFA, are risk factors for irritable bowel disease in humans. Although linoleic acid is considered an essential nutrient and required at 0.5 to 2% of total calories for proper development, North Americans can currently consume between 5 to 10% dietary energy from omega-6 PUFA. So there's quite a lot to unpack there. Essentially, the oiling of America, or the oiling of the West, was something that was brought on by the cholesterol heart disease hypothesis. And what they found was that the omega-6 polyunsaturated fatty acids actually lower the cholesterol inside the bloodstream, and cholesterol was thought to be the cause of heart disease. Obviously, that's not the case. There's a lot of research against that now. There's a lot more going on than just, you have high cholesterol. I've covered this in previous videos or touched on it a bit. I'll continue to cover it as I go through all of these studies. But what we're essentially seeing here is that omega-6 PUFAs have increased significantly in the diet, to levels that weren't previously eaten before. So the actual requirement for omega-6 polyunsaturated fatty acids in the research seems to be 0.5 to 2% of calories, whereas North Americans are currently consuming between 5 and 10% of dietary energy from omega-6 PUFA. So they're between 10 to 20 times more omega-6 PUFA than is necessary for the requirement. Now, why is this problematic? Well, the polyunsaturated fatty acids have negative effects. Both do. The omega-6 PUFAs are precursors for the inflammatory prostaglandins, which are types of molecules called eicosanoids. They're a type of eicosanoid. These basically cause the large portion of inflammation in the body, and linoleic acid is their precursor. Also, linoleic acid is extremely susceptible to being peroxidized or being damaged because of its double bonded structure. So while there may be a requirement, which is debatable the amount, and if there is a requirement, at least Dr. Ray Peat debates that there's an actual requirement for these fats. Even if there is, the requirement is extremely low, and it's extremely difficult to get below like 2 deficiency levels with a normal whole foods diet, or maybe not normal, but a whole foods diet. And this is from someone, myself, who's been trying to get polyunsaturated fat intake as low as possible, for quite a period of time, within reason, obviously. And I could cover that in another video on how I do that. But essentially, on a normal diet, you're going to be sufficient in omega-6. There's kind of no way to avoid it. If you're eating like 2 eggs in a day, there you go. Especially if you have, steak in there, maybe some butter, maybe some fruits, whatever the deal is. If you're having eggs, if you're having chicken, all of those types of things, you're going to hit your target, no problem. Now, you probably want to stay at those. What's more likely is you want to focus on lowering it rather than trying to get enough, because it is so pervasive in all of the foods. So again, very difficult to get into deficiency levels. The requirement is actually quite low, even by the research standards, perhaps even lower than what they state here. And then Dr. Pete argues if it's even required at all. And now what they're basically starting to find is that omega-6 PUFAs are increasing or associated with increased metabolic disorders, and then also associated with things like autoimmune diseases like irritable bowel disease. So now they're finding problems from this recommendation to increase large amounts of omega-6 fatty acids inside the diet. So anyway, the researchers continue. This is due to an increased, so the 5 to 10%, the North Americans' consumption of 5 to 10% of dietary energy from omega-6 PUFAs is due to an increased intake of omega-6 PUFA-rich oils such as sunflower, safflower, and corn. Also, soy would be in there as well as consumption of farm animals raised on seed oils rich in omega-6 PUFA. This is quite interesting. Essentially, and a lot of people don't think about it this way, but what your food eats, what the chicken and pigs or the pork and chicken that you eat, its consumption or its composition is dependent upon what those animals ate before they were slaughtered. So if you're feeding the chickens and the pigs large amounts of soy and corn, etc., then you're going to have a lot of soy and corn oils inside those animals. And ruminants like cows, goat, sheep, bison, you're not really going to see that, because their rumen basically saturates these polyunsaturated fatty acids or changes their structures to create trans fatty acids, like conjugated linoleic acid, which has actual health effects in opposition to linoleic acid. So you're often safer with the ruminant animals like cows, goats, sheep, and bison. So that's just an interesting side note there. In parallel, they continue, the consumption of fish oils rich in long-chain omega-3 PUFAs like docosahexaenoic acid or docosahexaenoic acid and eicosapentaenoic acid, which are responsible for numerous anti-inflammatory effects, and we'll get into the nuance of this here, represents only 0.15% of dietary energy in the North American diet. As a result of this imbalance, there has been interest in supplementing our diets with sources of omega-3 PUFAs such as fish oil pills, which have been suggested to prevent symptoms of IBD, irritable bowel disease. However, the effects of omega-3 PUFA supplementation on gut health are conflicting, with some studies showing they are beneficial, while others demonstrate they exacerbate colitis. So the reason they're bringing up PUFA here as percent is because there's an idea of ratios between omega-3 to omega-6. I think they talk about ideal being 3 to 1 omega-6 to omega-3. Now, you wouldn't want to ever get your omega-3 content to 5 to 10% dietary energy. That would be quite problematic from a taste perspective, and then also from a peroxidizability perspective, where the omega-3 fatty acids, specifically the long-chain ones from fish oil, are very easily peroxidized. Even though the shorter chain ones that you see with linolenic acid or alpha-linolenic acid, which is what you see in flaxseed, are also highly peroxidizable, to the extent that they have to be, a lot of these fats have to be stored, refrigerated, ideally, in amber bottles to protect them from the light, and then sometimes they even put nitrogen in the bottle to protect it from oxidizing spontaneously, even under those considerations. So they are, and the peroxidizability essentially means their fats are going rancid. They're having a damaged structure, and I don't know why people don't think that this is going to happen in the digestive system. So you're taking a fat, you're dumping it in a series of bile acids, and you're putting it in basic things, exposing it to enzymes, exposing it to bacteria, and you're also raising the temperature from whatever your refrigerator was set at to 98.6 degrees Fahrenheit or 37 degrees Celsius. So these fats are going to peroxidize in the intestine. If you're taking your fish oil with your steak, the heme iron from the steak is going to peroxidize it as well. So they're extremely liable to be damaged overall. While they may have some balancing effect from an inflammatory profile on the omega-6, you would never want them in high intake of the diet, and they're not even recommended in high intakes of the diet because of these known issues. Even the mainstream regulating bodies are talking about taking them at like high doses, like 3 grams in a day, which is, what's that, 3? So that's 18 calories worth of fat. So it's they're going to be in a low intake of the diet. Just by nature of all of these different things. And also, we're going to talk about here, but the anti-inflammatory effects, as I mentioned, are questionable in their mechanism. And then they're also talking about here how the omega-3 PUFA supplementation in the gut are conflicting. So we don't have a clear-cut answer on their benefit. As the researchers continue, they say in this study, we hypothesized that excess dietary omega-6 PUFA would increase colitis susceptibility through the microbe immunity nexus. C57BL6 mice were fed high-fat diet. So the diets, there was 3 diets. There was the 20% by weight diet that was corn oil. There was a 20% by weight diet that was corn and fish oil. There was 19% corn oil and 1% fish oil. It talks about in the methods on the bottom of the paper. And then there was a low-fat diet, which was kind of the control, which was 5% corn oil. Now, keep in mind, it's 5% corn oil by weight, so we're still not a low PUFA diet per se. We're just lower than the omega-3 corn oil mixture and then the pure corn oil mixture. They say here, we evaluated this in the intestinal microbiota, induced colitis with enteric bacterial pathogen Citrobacter rodentium, and followed disease progression. So they wanted to see first, what is the effect of these different diets on the intestinal microbiome? And then they wanted to see what happens to the intestinal microbiome, or what happens to these rats when they're exposed to this pathogen, Citrobacter rodentium, on these differing diets. And so they followed disease progression after. They're going to give us a little TLDR here, so too long didn't read. What they basically say is omega-6 PUFA enriched pro-inflammatory gut microbiota and exacerbated intestinal damage and inflammation during infection. Supplementing the omega-6 PUFA diets with omega-3 PUFA reversed these pro-inflammatory blooms and instead enriched anti-inflammatory microbes corresponding with less intestinal damage. However, omega-3 PUFA supplementation also reduced the presence of intestinal alkaline phosphatase expressing cells, resulting in the inability to detoxify lipopolysaccharide, aka endotoxin. The fish oil supplemented diet resulted in increased mortality during infection due to sepsis, evident by the presence of serum lipopolysaccharide binding protein, interleukin-15, and tumor necrosis factor alpha. So, what essentially went on in plain English? The omega-6 PUFA diet, the corn oil diet that was 20%, caused inflammation or created a pro-inflammatory microbiome and caused intestinal damage and inflammation during infection. So it just made things worse, made the infection worse as far as inflammation goes. The omega-6 PUFA diet with the omega-3 PUFA diet blocked these inflammatory changes and basically led to stopped the production of this pro-inflammatory gut microbiome. However, it also created a degree of immunosuppression that led to these rats supplemented the fish oil having increased mortality. More of these rats died, and then they also had increased markers of exposure to endotoxin. So omega-3 supplementation blocked the upregulation of these important cells, intestinal alkaline phosphatase expressing cells. The alkaline phosphatase is an enzyme that can degrade this endotoxin, the LPS, and so the PUFAs stop these cells from moving or being increased in the intestine in response to the infection. And so what happens is more of this endotoxin was able to make it into the bloodstream and induce sepsis, and then the sepsis was worse and it killed the rats. So it essentially immunosuppressed the animals, even though their intestines weren't as inflamed. So not a good thing overall. So on one side of the spectrum, you have omega-6, which is causing increased inflammation, making the inflammatory response worse, but not killing the rats or the mice here. Whereas the omega-3 is basically making the inflammation not so bad, but also, but then causing a immunosuppression that allows the rats to die from the infection. So it's on one side you're too much, and on the other side you're not enough. And we'll talk about what the extension is here. So as we get into the results here, the first thing they talk about is omega-6 PUFA-rich diets cause dysbiosis, while fish oil supplementation can reverse these effects. They start off here and they say omega-6 PUFA-rich diets enrich the microbiota with Enterobacteriaceae, which are associated with irritable bowel disease, and segmented filamentous bacteria, previously shown to induce responses that drive experimental colitis. So before the infection, the omega-6 PUFA-rich diets increased the microbiome with these toxic or these pathogenic bacterial species or phylums. And then, so that was the Enterobacteriaceae and then the segmented filamentous bacteria. Both of those associated with irritable bowel disease or inflammatory bowel disease, and then also experimental colitis. Then they say, in contrast, omega-3 PUFA supplementation prevented these specific enrichments, while both high-fat diets increased Clostridia species compared to the low omega-6 PUFA control, or essentially the low-fat diet control. The omega-3 PUFA supplemented group had reduced abundance compared to the omega-6 rich group. So the Clostridia species are specifically Clostridium coccoides group, which is considered an opportunistic pathogen, was increased more in the omega-6 than in the omega-3, but it was increased more in both of those higher fat diets than in the lower fat diet. So the control diet had less of that overall. They continue, and they're going to basically say what I just said. The omega-3 PUFA supplemented group had fewer microbes from the Clostridium coccoides group, which are opportunistic pathogens associated with inflammatory bowel disease. Besides the lack of potential pathobionts with omega-3 PUFA supplementation, this group also had enriched populations of beneficial microbes lactobacillus and bifidobacteria, known to be anti-inflammatory during murine colitis, which is just mouse colitis. Omega-3 PUFA supplementation also induced Enterococcus faecium, which has reported probiotic properties. In general, both high-fat diets reduced bacteroid species, which led to a trend associated with obesity, and correspondingly both groups of mice were similarly obese, data not shown. So the mice were obese from this, from the high-fat feeding, the 20% by weight. They didn't show that data, and then that also correlated with a decrease in the bacteroid species. That's like another association that they talk outside of other studies where I think it's increased firmicutes and decreased bacteroids leads more towards obesity. Besides that, what they're essentially showing here is that the omega-3 bacteria, or the omega-3 PUFA group, had increased commensal bacteria, lactobacillus and bifidobacteria, and less pathogenic bacteria. So the omega-3 group is creating a microbiome, or has a microbiome that's less pathogenic overall than the omega-6 group. That is actually a good thing. However, some of the other effects that we're going to dive into are not a good thing. And the other thing that we'll talk about is, and we'll get to this in a bit, you can modulate the content of lactobacillus and bifidobacteria in the gut without having to take omega-3s. So if omega-3s have a toxic or negative effect or immunosuppressive effect themselves, while they do have a beneficial effect modulating the gut microbiome, if there's other things that can have that effect modulating the gut microbiome, why not use those things, especially if they don't have this negative effect that the omega-3 does? So that's essentially, we'll dig into that a bit. Last thing they say here is, overall, while omega-6 PUFA-rich diets induce the growth of microbes known to induce pro-inflammatory responses, omega-3 PUFA supplementation reversed this, and in parallel enrich beneficial bacteria. I think that's actually the best thing that came out of the omega-3 piece here. Everything else is kind of not good. So as we get into it here, they say omega-6 PUFA-rich diets, this is the next title of this result section, cause severe colonic damage, and while omega-3 PUFA prevents this damage, it leads to increased mortality during Citrobacter rodentium infection. So again, I'm not going to read everything here, but the normally resistant C57BL/6 mice fed omega-3 PUFA supplemented diets suffered increased mortality during infection, where 30% of the mice had to be sacrificed by day 8 post-infection, while both low and high omega-6 PUFA diets resulted in similar weight changes during infection. The omega-3 PUFA supplemented group suffered the greatest weight loss throughout days 5 to 10 post-infection, despite similar pre-infection body weights and caloric intake in mice fed both high-fat diets. So, very important to point out here, the omega-3 supplemented group had significantly more mortality than the omega-6. So this strain of mice, the C57BL6, are resistant to this infection as far as mortality goes. Yet when they were supplemented the omega-3 fatty acids, they actually died. So 30% of them had to be sacrificed because they were that sick 8 days after being infected. Whereas in the omega-6 group, you didn't see that. The omega-6 rats weren't dying, they were just more inflamed. So this is pointing again to an immunosuppressive effect, which is not good. The other thing is the omega-3 supplemented rats had a severe loss in body weight, or suffered a greater loss in body weight than omega-6 and the low-fat fed rats. This, I think, correlates with the tumor necrosis factor alpha, which was higher in the omega-3 rats. In other studies, even in humans, high amounts of tumor necrosis factor alpha is strongly associated with cachexia. Cachexia is a wasting syndrome. It's essentially like you're just burning through all your energy, you're eating away your tissues, and this can be induced by the tumor necrosis factor alpha. They actually treat it with, I think, tumor necrosis factor alpha blocking drugs. And the reason I think this happened here is because the rats in the omega-3 group were immunosuppressed. So when the endotoxin hit the bloodstream, it was highly active, and there was more of it in the omega-3 group, which raised tumor necrosis factor alpha. Endotoxin raises tumor necrosis factor alpha, and then that led to like a form of cachexia to some extent in these rats, or a wasting of their weight, which is not usually a good sign at all in sepsis, in cancer, in any of these types of really acute situations. So not good overall, really not good. Furthermore, they say yet during infection, the omega-3 PUFA fed mice displayed similar histopathological severity based on mucodepletion, hyperplasia, immune cell infiltration, edema, and epithelial integrity as to the low omega-6 PUFA control. So their intestines look good. They were similar to the omega-6 PUFA control, the low omega-6 PUFA control. So that's the 5% by weight corn oil diet. They were similar, so the omega-3 had an effect minimizing the inflammation, but they still died more. So they just died with nicer intestines. I mean, that's not a better outcome. They say, in contrast, omega-6 PUFA rich fed mice displayed the most severe histopathology. We examined intestinal epithelial cell death to determine the effect of PUFA diets on intestinal homeostasis, since this correlates with intestinal damage. Both high-fat diets induce cell death prior to acute colitis, revealing that epithelial cell homeostasis was disrupted by high fat alone. Following infection, mice fed omega-6 PUFA-rich diets had increased colonic cell death, whereas omega-3 PUFA supplementation attenuated this response, indicating that a high omega-6 to omega-3 PUFA ratio induces exacerbated colonic damage. So what are they saying here? Before the rats were infected with the Citrobacter rodentium, the high-fat diets, the 20% by weight diets alone, cause a disruption inside the homeostasis of the epithelial cells in the intestine. So those are the cells lining the lumen of the intestine. These are the cells exposed to the lumen. So the high-fat diets cause problems here. And then following infection, the omega-6 PUFA-rich diets had increased colonic cell death. So it was their destruction of the cells lining the lumen of their intestine was worse. And the omega-3 PUFA was able to block this response to some extent. So again, the omega-3 PUFA has a less inflammatory, or it helps to deal with the inflammation from the omega-6. Again, because the omega-3 group was 19% corn oil, 1% fish oil. So even just a little amount was helping to minimize the inflammation. But as you know, even that 1% was causing an increased mortality where you had 30% in the omega-3 group, which is insane. The next result here, they say, is omega-6 PUFA-rich diets increase intestinal inflammatory cell infiltration, Well, omega-3 PUFA supplementation impairs infection-induced immune responses. So they say omega-6 PUFA-rich diets enrich pro-inflammatory inducing microbes and worsen colitis, while omega-3 PUFA supplementation reverse these effects and enrich beneficial microbes. We know that. We just talked about that. We examined colonic neutrophil and macrophage cell infiltration and found that omega-6 PUFA-rich diets increase infiltration of F4/80 macrophages or F4/80 positive macrophages and myeloperoxidase neutrophils compared to the low omega-6 PUFA group during infection. Myeloperoxidase is an enzyme that these cells use to basically attack bacteria to create reactive oxygen species and destroy bacteria. The F4/80 macrophages are a type of macrophages. Macrophage basically gobbles up, or it's like a Pac-Man you can think of, that eats these pathogenic bacteria. So they basically want to see, what's going on with the cell infiltration at the intestine. Are these immune cells moving to the intestine? And then what's the effect? The omega-6 diet, omega-6 PUFA-rich diet, increased infiltration, and then the low omega-6 PUFA, or compared to the low omega-6 PUFA-rich group. In contrast, omega-3 PUFA supplementation restored immune cell infiltration similar to the low omega-6 PUFA control. We also examined the levels of prostaglandin E2. This is an inflammatory marker, a key inflammatory marker in the gut, and found that mice fed omega-6 PUFA-rich diets had the highest infection-induced levels of colonic PGE2. Not surprising, because omega-6 PUFA is a precursor to the formation of the prostaglandin. So it's increasing these inflammatory signaling. Omega-3 is actually blocking this, and the other thing you're seeing is that there's large amounts of, essentially, the high omega-6 in the diet overall, or in the tissues, is when you have an immune response, it's like having a bunch of dynamite stacked up in your tissues. It's going to trigger a large explosion or a large signaling of an inflammatory response. The omega-3 is blocking this signaling to some extent, or to a large extent, and in multiple ways. And so that's basically the body is not getting the signal in the omega-3 group, hey, we have a problem here. Whereas in the omega-6 group, you're having the signals way too strong. It's like, oh, we have this massive problem here. Maybe if the problem isn't even that massive. And then in the low fat group, the 5% corn oil group, which it's still corn oil again. In that group, what you're seeing is that the signal isn't so strong, but it's still present. So the omega-3 group is dampening the signal way too much for the immune response. The omega-6 is amplifying the signal way too much. And in the lower fat group for these mice with the 5% corn oil is not creating such a massive signal that it's immunosuppressing them. They're still getting some inflammation and dealing with the infection, but they're not dying. So that's kind of like the middle ground here. I think that there's even a better middle ground, but we'll get into that. Here's some graphs here just showing the enrichment of the different bacteria inside in the microbiome of these rats. So here we see increased Enterobacteriaceae for the high omega-6 PUFA group. So this is an inflammatory bacteria. Same thing with the segmented filamentous bacteria, and the high omega-6 group is being enriched. It's also slightly enriched more so in the omega-3, omega-6 PUFA group for Enterobacteriaceae and segmented filamentous bacteria here than it is for the lower fat group. So both the 20% fat by weight group cause an increase in this in these more pathogenic types of bacteria, but it was worse in the just straight corn oil groups. Same thing with Clostridia. You see an increase in the Clostridia, specifically the Clostridia coxoides groups, or they're saying the Clostridia coxoides group down here, but there's the increased Clostridia species in the omega-3 supplemented group and in the pure high-fat corn oil group, and you're not seeing so much of that inside the lower fat group. Now, in the omega-3 group, you're seeing a much higher bifidobacteria and lactobacillus, and then you're also seeing a higher Enterococcus faecium, so you're seeing increase these beneficial species. You actually aren't having much of those in the lower fat group. And then the Clostridia coxoides highest in the low PUFA group, and the bacterioids, which is actually a beneficial species, or at least based on what they're talking about in this paper, as being higher amounts of this being associated with lower obesity, and then Enterococcus faecalis. So you're seeing higher bacteroids, Enterococcus faecalis, in the low PUFA group, the 5% corn oil diet, versus the high PUFA, which is 20% corn oil, and then the 19% 1% omega-3 group. So this is what they're talking about. So the omega-3 had like increased some of the beneficial species. The omega-6 increased the negative species, and then the low-fat diet was kind of didn't really, it was kind of in the middle again. It didn't increase the negative species. It didn't increase necessarily the beneficial bacterial species in the microbiome. They say, in contrast, omega-3 proof of supplementation did not induce prostaglandin E2 cell infiltration during infection, suggesting that these mice were impaired in mounting an inflammatory response to infection. So this is, again, going back to the prostaglandin E2 with the signaling. The omega-3 is blocking that signaling. So you're seeing that that mice were impaired in mounting an inflammatory response to infection in the omega-3 group, in that 1% omega-3 group. As they continue to say, in general, and let me see if I can pull up the prostaglandin E2. This one is the histopathology. I'll go over that once I finish this section. And so I'll go over those once I finish this section. Next thing they say, in general, the omega-6 PUFA-rich fed mice were able to mount an inflammatory response evidenced by the induction of cytokine expression post-infection. In contrast, the omega-3 PUFA supplemented group was unable to induce such responses during infection, evident by the lack of induction of interferon gamma, tumor necrosis factor alpha, IL-17 or interleukin-17 alpha, interleukin-22, interleukin-23, as well as the chemokine realm beta, compared to pre-infection expression. This data reveals that omega-3 PUFA supplementation impairs infection-induced cytokine responses, even though omega-3 PUFA supplemented mice had increased interleukin-23 and realm beta expression under uninfected conditions. So the omega-3 PUFA changed the expression when they didn't have the infection, but when they did have the infection, it altered some of the responses here to the infection. Now, there's multiple time courses here that we're discussing, so I'll get into that in a little bit. Before I go on to this next section, I want to just go over these graphs here for you guys. So this is where they're talking about the survival. So in graph A, what you're seeing here is 100% survival, and then you get down to 70% survival. So you can't see that the low omega-6 PUFA and high omega-6 PUFA are together on this line because they plotted them together, so you can't see the triangles underneath the squares. But basically what you're supposed to see here is that low omega-6 PUFA and high omega-6 PUFA, all the rats or all the mice were alive at 10 days post-infection, but the omega-3 group, 30% of them died. So at 8 days, 30% of the rats had died from the infection. In the other two groups, you didn't see any death. Other thing you're seeing here is that the omega-3 group had that wasting effect. So by 10 days post-infection, they lost 20% of their body weight, which is a huge amount of body weight. So that's if I'm a 200-pound person, I just lost 40 pounds, which is that's a lot of weight to lose in 10 days. Although rats' metabolisms, to be fair, are different than human metabolisms. Next thing we're going to talk about is histopathology score. Basically, what they're showing here is those factors like epithelial integrity, inflammatory cell or immune cell infiltration, etc. All of those types of things, they create a score here. The histopathology between the low omega-6 PUFA and the omega-3 PUFA after infection was about the same. The high omega-6 PUFA had worse histopathology score, so the tissue structure was more damaged. The other thing, so this section is looking at the prostaglandin E2, the myeloperoxidase, and the macrophages. So what you're seeing is in the low PUFA, the 5% corn oil diet, and then the 5% or the 19% PUFA, 1% omega-3 diet. These red areas are the staining of the macrophages, and so what you're seeing is, or let me just make sure for sure that that's the macrophages. So yes. So this is the staining of the macrophages. So what you're seeing is a higher amount of these macrophages in the high omega-6 group. You're seeing really not so much macrophages in the omega-3 group, and then you're seeing marginal amount of macrophages in the low fat, the 5% corn oil group. So really, you're seeing this again, amplified signal, blunted signal, more normal signal. Again, it's 5% corn oil, so we can't do anything about that. And then this graphs are showing the same thing. Here, the myeloperoxidase, again, is that enzyme that the neutrophils use to attack bacteria by producing ROS, you're seeing this amplified signal in that high omega-6 group. You're seeing some signal to some extent in the low PUFA group, the 5% corn oil group, not that much. And in the omega-3, you're not really seeing, again, it's very minimal signal from the myeloperoxidase expressing neutrophils, or myeloperoxidase positive neutrophils. So again, you're seeing block signal in omega-3, amplified signal in omega-6, kind of normal signal in the lower fat group with these mice. Last one here, interesting one. Prostaglandin E2, the inflammatory marker. This one we can really see in the high omega-6 group, really high amounts of that prostaglandin E2. The tracers are on the intestinal epithelium here are lit up. On these other groups, not as much. So you can see that this high omega-6 group is really saturated with this inflammatory molecule. Obviously, a lot of inflammation is not ideal. So this is not necessarily a good thing. And that's, again, the omega-6 PUFA from corn oil, linoleic acid, is a precursor to the prostaglandin. So I'm not surprised. Let me just make sure. Let me go down here and see if there's... Nope, so we haven't gotten to those yet. Perfect. Next section they go into, they say omega-6 PUFA-rich diets resulted in increased translocation of gamma proteobacteria. So they start here. While few microbes were present prior to infection, they were found in the submucosal region of the mice-fed omega-6 PUFA diets, while none were present in the mice-fed diet supplemented with omega-3 PUFA. Because Citrobacter rodentium is a member of gamma proteobacteria, we could also use this probe to examine the localization of the pathogen during infection. We found the microbes deeply invading the crypts, and were also observed in the submucosal region of colons from mice-fed omega-6 PUFA-rich diets. So basically, this type of I think it's a phylum, class or phylum. What did they say here? They didn't tell us which one. Essentially, the gamma proteobacteria, of which Citrobacter rodentium is a part of, was getting across the intestinal barrier from the inflammation in the omega-6 group. So the omega-6 group, the high omega-6 group, was causing inflammation, and then the bacteria were able to move across the intestinal barrier and invade the crypts and invade the underneath the mucosal layer of the intestine. So that's not a good thing at all. You don't want basically the intestinal barrier is supposed to keep these things out. So the inflammation from the omega-6 is impairing this process. In contrast, they say the mice-fed omega-3 PUFA supplemented diets were able to contain the pathogen at the top of the crypts, and bacteria were not observed in the submucosal region. This suggested that the pathogen was more systemic in the mice-fed omega-6 PUFA-rich diets, but that fish oil supplementation prevents this bacterial translocation. So this is actually not necessarily a terrible thing, but the bacteria, I'm assuming because there was less massive amounts of inflammation, the bacteria weren't able to get deeper into the tissues. The tissues still had their integrity in the omega-3 group, largely because of this immunosuppressive response. Whereas in the omega-6 group, that large amount of inflammation was kind of tearing up the intestinal epithelium, allowing bacteria to get through. They also say we found an increase in colony-forming units in the tissues from mice fed omega-6 PUFA-rich diets, which was reduced following omega-3 PUFA supplementation. So they talk about this in the lymph nodes and the spleen. Essentially, the bacteria that get across the intestinal epithelium, the intestinal barrier, are going to be picked up by immune cells, transported to the spleen, which is like the hub for the immune system to some extent, and also the lymph nodes. And that's been essentially what they found. So if you get past the intestinal barrier, well, the immune system is still there to kind of clean up the mess. So that's what you saw in the omega-6 rich diets. In the omega-3 PUFA rich diets, you're not seeing that translocation to these other tissues because of this immunosuppression. Or at least my hypothesis from reading the papers from this immunosuppression. Next here is the next title is omega-3 PUFA supplementation induces sepsis in Citrobacter rodentium infected mice. I say, in support of the observed translocation of Citrobacter rodentium across the mucosal surface in the high omega-6 PUFA diet, we found lipopolysaccharide binding protein in the serum of these mice prior to infection. So this protein is elevated to pick up endotoxin and detoxify it and protect the body from it. So if the bacteria are moving across the intestinal epithelium, or the intestinal barrier, and they're showing their endotoxin to the immune system is going to upregulate this lipopolysaccharide binding protein to protect the tissues from the bacteria. It's an immune response. They say, in contrast, there was no induction of lipopolysaccharide binding protein in mice fed omega-3 PUFA supplemented supplements prior to infection, but during infection, lipopolysaccharide binding protein levels increased. So they're saying this goes hand in hand with the omega-6 PUFA kind of damaging the intestine, allowing bacteria through. Before the mice were actually infected with Citrobacter rodentium, this was causing this pathogenic bacterial. The omega-6 PUFA was causing a pathogenic bacterial profile in the microbiome and also allowing those bacteria to get through the intestine, whereas the omega-3 was not. And that reason you're seeing the lipopolysaccharide binding protein upregulate is, again, because it's a protective effect against the endotoxin produced by these pathogenic bacteria. Furthermore, they say, despite there being lower levels of pathogen in the spleen, and I think MLN is let's see where they put it. They have it down here. MLN is mesenteric lymph nodes. So the mesenteric lymph nodes are the lymph nodes of the intestine. Spleen and the mesenteric lymph nodes in the omega-3 PUFA supplemented group. These mice had the greatest induction of serum IL-15 and tumor necrosis factor alpha. So in the omega-3 group, they had large amounts of tumor necrosis factor alpha, and they had less pathogen in the spleen. So the bacteria weren't getting through the intestine like the inflamed intestine of the omega-6 fed rats. But in the omega-3 fed rats, so they weren't getting across, but the endotoxin that was getting across wasn't being mopped up or detoxified as well, so it's causing these large spikes in interleukin-15 and tumor necrosis factor alpha, which I think is what induced that cachexia-type symptoms, and then also led to the increased sepsis and mortality. What they continue to say is these results suggested that while omega-6 PUFA-rich diets resulted in increased bacterial translocation under uninfected conditions, they were able to avoid infection-induced sepsis. In contrast, omega-3 PUFA supplementation inhibited bacterial translocation in the absence of infectious insult, but these mice suffered sepsis following Citrobacter rodentium infection. So omega-6 allowed bacteria to go across the epithelium by causing, I think through the excess inflammation, but because they were signaling all this inflammation and ramping up the immune system was still able to deal with the infection. So the mice weren't dying from the infection; they were just not having a good time. In the omega-3 group, the mice, the infective bacteria weren't necessarily getting so much across the intestine, but the things that were getting across the intestine weren't being picked up by the immune system because of the lack of the signaling due to the blockade from the omega-3s. So essentially the mice went into sepsis, so they weren't able to protect themselves, and then they were dying, which is, you're in one case you're feeling what's going on and it kind of sucks, and in another case you're not feeling what's going on and it doesn't suck as much, but you're dying from it. So it's like in both cases it's obviously not an ideal situation. Clearly there's a middle road here. Furthermore, they go into intestinal alkaline phosphatase expression and lipopolysaccharide dephosphorylation, or essentially degradation or detoxification. You can put that word in here instead of dephosphorylation. Was impaired in mice fed omega-3 PUFA supplemented diets during infection. They say, we examined the expression of intestinal alkaline phosphatase, a key endogenous mucosal defense factor inducible by microbiota. Intestinal alkaline phosphatase dephosphorylates or detoxifies lipopolysaccharide during infection, preventing sepsis. We found that the greatest number of intestinal alkaline phosphatase positive cells infiltrating in the colonic submucosae from mice fed omega-6 PUFA-rich diets. So the omega-6 is, again, amplifying that signal. In contrast, mice-fed diets supplemented with omega-3 PUFA had fewer intestinal alkaline phosphatase positive cells. Since intestinal alkaline phosphatase detoxifies lipopolysaccharide through dephosphorylation, so it basically removes phosphate groups from the lipopolysaccharide, we examined the ex vivo lipopolysaccharide phosphorylation activity of colons for mice-fed PUFA diets, similar to Goldberg et al. While both low and high omega-6 PUFA-rich diets induced LPS dephosphorylating activity during C. rodentium infection, the omega-3 PUFA supplemented diet resulted in an impaired ability to dephosphorylate LPS during infection. Overall, these results show that omega-3 PUFA supplementation reduces pathogen translocation, but also impairs intestinal alkaline phosphatase expression and the ability to dephosphorylate lipopolysaccharide during infection. As a result, any pathogen that escapes across the barrier in omega-3 PUFA supplemented mice may be more toxic, leading to increased systemic inflammation associated with the increased mortality during C. rodentium infection. So we just talked about this. The omega-3 doesn't cause this inflammation that irritates the intestine and allows bacteria to get through. But whatever does get through, then since the system is immunosuppressed to some extent by the omega-3s, it's going to have a huge impact. It's going to have like a really big impact on the system. And that's why you're seeing the sepsis and increased tumor necrosis factor alpha, etc., going on with these rats. And here they're just showing the inflammatory markers in the different groups here. I'm not going to go through these specifically, but you're seeing the omega-3 PUFA group is lowering these factors except for tumor necrosis factor alpha. And then, let's see. So non-infected. In some of the non-infected groups, the omega-3 supplementation was actually higher for some of these factors, even though they didn't get as high following infection. So that's it for the results. We're going to go into the discussion. And basically what they say here to start, an increase in IBD incidence has been associated with increased dietary intake of omega-6 PUFA in humans. Well, inflammatory bowel disease patients remain in remission longer when dietary intake of omega-3 PUFA increases. Not surprising if it has an immunosuppressant effect, and most of the treatments for inflammatory bowel diseases and autoimmune diseases are immunosuppressants. So yes, it's going to increase remission, and then high intakes of omega-6 PUFA will clearly make it worse because they're going to amplify this inflammatory response. And it looks like high intakes of omega-6 PUFA induced the inflammatory bowel disposition anyway by changing the bacteria in the microbiome and then also upregulating inflammatory responses to them inside the intestine. So the omega-6 may even be a contributing factor. That's why you're seeing IBD is associated with increased dietary intake of omega-6, which we're now at 5 to 10% because of all the corn, soy, and safflower oil in the diet, etc. So let me cover these images here really quick. What they're showing is the movement of the pathogen in the different groups. So in uninfected and infected, there's an uninfected group and an infected group. This staining here is of the pathogen Citrobacter rodentium of the gamma proteobacteria. What you're seeing, so this is the high omega-6 PUFA and omega-3 PUFA group. So this is the omega-3 group here. What you're showing in the infected side is that the bacteria aren't in the crypts. These are the crypts. So the spaces between the villi and the intestine, they're all kept kind of in the lumen. On this one here is the high omega-6. You can see that the bacteria are starting to infiltrate into the crypts in between the microvilli and the villi of the intestine. And then here's the submucosal layer on the right side here. So this is a deeper magnification. And you're seeing the bacteria able to get to the submucosal layer. And the omega-3, you're not seeing that. Then inside the low omega-6 PUFA, again, you're seeing that the bacteria aren't penetrating into the crypts here inside the intestine. They're not getting between the villi. They're staying in the lumen, and they're also not in the submucosal layer. So the high omega-6 is allowing the bacteria to get to the submucosal layer and invade in between the microvilli, whereas the low fat and then the omega-3 is blocking that from happening. And then they have a negative control. They also looked at colony-forming units. This one here is the mesenteric lymph nodes, and then the spleen. In the spleen, you're seeing lower colony-forming units in the omega-3, and then lower colony-forming units in the mesenteric lymph nodes, and then you're seeing higher amounts of these bacteria. So colony-forming units are the bacteria. Excuse me, should have said that initially. The bacteria are highest in the spleen in the high omega-6, and highest in the mesenteric lymph nodes in the high omega-6. And the low fat, you're seeing a little bit more than the PUFA, probably because the PUFA has essentially the immune cells are bringing this bacteria to the spleen and to the mesenteric lymph nodes. So with the high omega-3, if the high omega-3 is causing an immunosuppression, then what may be going on is that the immune cells aren't picking up the bacteria and bringing them to the spleen and the mesenteric lymph nodes. So you may see some immunosuppression here, which may make it lower, because as you looked at the pictures above with the staining, the low-fat diet also didn't have a lot of bacteria in the submucosal layer, and it also didn't have a lot of bacteria in the crypts. So this lower amount, to some extent, than although it's, maybe it's in the spleen, it's significant, although they're not showing significance between the omega-3 and the low-fat. So maybe they're not a significant result, but the trend to have the lower bacterial count in the mesenteric lymph node in the spleen may be due to immunosuppression from the omega-3 compared to the low-fat. And then the higher amounts in the omega-6 are most likely due to the inflammatory response in the intestine, allowing these bacteria to get through. So here we go. They talk about, here's some interesting graphs going through the post-infection, the amount of these cytokines inside the serum. And as you can see, tumor necrosis factor alpha is off the chain here with the omega-3 group, what I discussed before. IL-15 is off the chain, and then lipopolysaccharide binding protein. So that endotoxin and those bacteria that are getting across the intestine, even if they're getting less so in the omega-3 PUFA group, they're having a much stronger effect because of the immunosuppression. So these cytokines are really getting upregulated. So quite interesting. We talked about that above. I'm just going to finish reading some of the discussion pieces here. They continue to say here in the discussion, they say, however, several colonic responses during infection were severely impaired. So supplementation with fish oil, which contains omega-3 PUFA, reverses these effects. Those are from the omega-6. However, several colonic responses during infection were severely impaired, including immune cell infiltration, cytokine-chemokine induction, and LPS dephosphorylation as a result of reduced intestinal alkaline positive infiltrating cells. Consequently, mice-fed diet supplemented with omega-3 PUFA suffered from increased infection-induced mortality associated with sepsis-related serum lipopolysaccharide binding protein, interleukin-15, and tumor necrosis factor alpha. Those are the graphs I just went over. They're saying the same thing here. You're seeing immunosuppression. This is largely looks like an immunosuppressive profile, right? We're having decreased immune cell infiltration to the sites of infection. We're having a decreased production of cytokines and chemokines, and we're having a decreased breakdown of toxic endotoxin, and then a reduction of the amount of intestinal alkaline phosphatase positive cells migrating to the site of infection. So you have this infection going on in the intestine, and then the cells are basically, the tissues aren't saying, hey, immune cells, come over here, we have a problem. And because the omega-3s are blocking that. The omega-6 are saying it too loud. You have like a bunch of people like, yo, you need to get over here. And then essentially once these, so when I was talking about time course before, once these, the inflammatory bacteria and then the endotoxin get across the intestinal epithelium, what you're seeing is that their inflammatory products are in the omega-3 group. So even though less is getting across, once they do get across, they're strongly signaling lipopolysaccharide binding protein, IL-15, and tumor necrosis factor alpha to be like, it's like the alarms went off too late. It's like the system has already been infiltrated by the spy, and it's like we have a spy and everything goes to lockdown. And so it's like causing this massive negative response. So it's really not good. Next thing they say, we found that diets rich in omega-6 PUFA produce microbiota with increased numbers of potential pathobionts, including bacteria from the family Enterobacteriaceae. This is similar to IBD patients and mice undergoing dextrin sodium sulfate colitis, supporting the hypothesis that omega-6 PUFA diets increase intestinal microbes which drive colitis. We also found these pro-inflammatory microbes were present in the colonic submucosa. And while we did not observe overt colitis prior to infection, we did find presence of serum lipopolysaccharide binding protein. This suggests that omega-6 PUFA diets or rich diets prior to infectious insult promote inappropriate inflammation. So before they even got infected, the omega-6 PUFA diets are moving towards intestinal inflammation. They're directly at the intestine, and then also changing the microbiome to be pathogenic. And you're also seeing an increased leakiness of the intestine with the omega-6. So it's doing three things. Even though they're not dying from the infections, it's increasing the inflammatory response. It's increasing the movement of these bacteria across the intestinal epithelium or intestinal barrier, and then it's increasing the amount of pathogenic bacteria. So 3 very negative things overall from omega-6. Now they get here to omega-3. They say, remarkably, diet supplemented with omega-3 PUFA prevented the bloom of Enterobacteriaceae, as well as the translocation of bacteria into the submucosal region, and instead promoted the enrichment of lactobacillus species and bifidobacteria species, both of which are associated with milder dextrin sodium sulfate and C. rodentium-induced colitis. Omega-3 PUFA supplementation prevented pathogen translocation, which may be due to the increase in lactobacillus species and bifidobacteria species, since both are known to increase barrier function. So the omega-3 group is not producing or protecting from the omega-6 bloom of these pathogenic species, and then it's protecting the intestinal epithelial barrier by upregulating lactobacillus and bifidobacterium. So essentially, it's modulating the microbiome. That is the benefit here, is the modulation or the modulation of the microbiome, not necessarily this anti-inflammatory effect of the omega-3 PUFA. Perhaps the anti-inflammatory effect plays a part in that microbiome modulation, but it seems like its anti-inflammatory effect is actually immunosuppressive, so it's actually causing problems. So the solution here, from my perspective, would be, let's find other ways to increase these beneficial bacterial species, aka fruit and veggie fibers, polyphenols, etc. Those will also increase the bacterioid species, which were mentioned to be positive in this side of the study. Let's minimize the immunosuppressive omega-3. And let's also minimize an extremely inflammatory omega-6. Let's move towards monounsaturated and saturated fats. And let's make sure that we have adequate tolerated fibers, polyphenols, plant compounds, fruits, vegetables, etc., herbs, to modulate the microbiome and avoid these problems. All the omega-3 PUFA and the omega-6 PUFA are both problematic, at least based on this study. And if the benefit from omega-3 is largely through modulation of the gut microbiome, increasing lactobacillus and bifidobacteria, well, we can do that without omega-3. So that would be my solution here, especially because the low PUFA group, which was considered the control, the 5% corn oil diet, was on par with the beneficial effects of the omega-3 without the immunosuppression and sepsis and death. So you can get the benefit without having to take omega-3. It just also includes not having large amounts of omega-6. So they conclude this study here. They say, our results suggest that omega-3 PUFA supplementation to an omega-6 PUFA-rich diet results in a milder colitis, but impairs infection-induced inflammatory responses important for preventing systemic Citrobacter rodentium infection. Similarly, another study demonstrated that mice fed high levels of omega-3 PUFA had impaired immune function and could not produce a response against Helicobacter hepaticus-induced infection. So they're seeing it with other bacteria as well. Consumption of omega-3 PUFA-rich diets may have anti-inflammatory properties. However, this may prevent the body from mounting appropriate immune responses critical for host defense. And following with this, the mice fed omega-3 PUFA supplemented high-fat diets suffered increased mortality during Citrobacter rodentium infection, while the lack of colonic inflammatory response likely played a role. A contributing factor was the inability to detoxify endotoxin, which is a major toxin, through the lack of phosphorylation activity in response to Citrobacter rodentium infection. In addition, there's a lack of intestinal alkaline phosphatase positive submucosal cells infiltrating into the colons of these mice. Intestinal alkaline phosphatase prevents sepsis by dephosphorylating lipopolysaccharide. While the pathogen did not translocate across the intestinal barrier at an increased level in the mice-fed diets with omega-3 PUFA, C. rodentium itself induces barrier dysfunction. Thus, some pathogen became systemic, and this resulted in sepsis. In support of this, mice-fed fish oil supplement diets had elevated serum-associated lipopolysaccharide binding protein, a clinical biomarker of sepsis associated with mortality. So that's really and here you can see that's really the gist of the study. I'll show you guys this really quick. This is the intestinal alkaline phosphatase producing cells. You can see we have a lot in the omega-6 PUFA, which is basically signaling, hey guys, come over here. In the low-fat diet, we see some to deal with the endotoxin, but we're not seeing really much at all inside the omega-3 PUFA, which is quite problematic. And then you're seeing a decrease in the LPS phosphorylating activity of the omega-3 group. So the low omega-6 group, the low-fat group, and then the high omega-6 group, the activity of their intestinal alkaline phosphatase was working well, but in the omega-3 group, it's had decreased activity, and then as we see above here, decreased amount of these intestinal alkaline phosphatase positive cells. There's one other thing I wanted to touch on. Oh, in the methods section here, what they talk about as far as supplementation, they say the diet oil components include corn oil, which is the high omega-6 PUFA diet, and corn oil plus fish oil, which is the omega-3 PUFA diet. For omega-3 PUFA supplementations, we follow the guidelines set by American Heart Association, recommending 0.5 to 1.8 grams of long-chain omega-3 PUFAs per day to offset high omega-6 PUFA diets, as EPA plus DHA are 34% of fish oil by weight. 0.5 grams of EPA plus DHA is available in 1.5 grams of fish oil. So they supplemented the fish oil in these diets at 1% to match the recommendations from the American Heart Association. And, I know this is a mouse study, but it's just kind of funny because it's like the American Heart Association recommendations here in mice led to 30% increased mortality during infections. So just something to think on there, although, again, this is in mice. So, yeah, that's about it for this study here. I would highly consider looking into your fish oil supplements if you are taking them and understanding their mechanism of action here. And then if you're able to recreate the mechanism of action in a better way, I would recommend avoiding them. I'm not opposed to seafood personally. I think seafood is great. I just am opposed to eating like 10 grams of omega-3 per day from seafood. I think that can be quite problematic. I think there are other beneficial effects of seafood. Some less well-known one is something called furin fatty acids, which I've talked about before with Hans, but I'll go through that a bit in another video. And yeah, so if you like this video, if you want to see more content like this, if you want to see more studies, if you have studies for me to go over or go through, I'd be happy to do that. You can drop a comment in the video, or you can find me on the Telegram group. I will drop a link inside the description section here. Hope you guys enjoy the study. Looking forward to see you again soon.